That brings us to chapter 11, where we are going to talk about the effects of solar radiation on the skin. And most of the chapter is going to focus on the effects of UVA and UVB radiation on the skin. But we will also talk about the effects of visible light and infrared light on the skin, as that has become a very important area of research in the last few years. And actually, several multinational companies are doing a lot of work in that area and are developing products that will fight against the effects of visible and infrared light. And as you will see, these actually cause free radical type damage in the skin. So we'll first talk about the interaction of light with the skin, and then we'll go on to talk about different chromophores in the skin. And one of those chromophores that we'll discuss is trans-Urocanic acid. So when you think of the chromophores, chromophores are molecules that absorb light, and when things in skin absorb light, usually there are several options on what can happen to those molecules, but most of the time they undergo some type of change or degradation, and this leads to some sort of damage in the skin. So trans-Urocanic acid is one of those, and the reason that we're going to mention that is that because trans-Urocanic acid is converted to cis-urocanic acid when it's exposed to UV light, and this actually causes what is known as UV-induced immunosuppression. Please note that trans should be italicized here. We'll also talk about some of the positive effects of sun or solar radiation, and that is in the production of vitamin D. And we'll show you how vitamin D production in the skin can actually lead to increases in the absorption of calcium in the gut. We'll also discuss UV-induced DNA damage, not only from a standpoint of UVB light, but also with the effects of UVA light. And then, of course, we'll discuss a little bit UV-induced immunosuppression. This is a very important area in skin research, as it is known now that there is a definitive link between UV-induced immunosuppression and photocarcinogenesis, which we'll also discuss in this section or this chapter. And then finally, we'll end up with some comments on the effects of infrared and visible light on the skin. So when we talk about the interactions of light with skin, we want to know, what are the light rays doing? And usually you have a couple different options. The light can basically come down, interact with the skin, and it can be reflected off of the front or the top surface of the skin, although this is only about 5% of the original incident light. So there isn't that much that's directly reflected. If skin looks really shiny, like if you put a oil on the skin, then you have higher reflectance occurring. But normal skin in its normal state, you won't have that much reflectance from the top layer. So a lot of the light actually goes into the skin structure. You can see it's passing through the stratum corneum here. There will be some absorption in the stratum corneum, especially if you have beta carotene present, for example. When you have skin, especially you'll see this in children's skin, because they eat a lot of baby food that's filled with carrots, and carrots have carotenoids in them. So they'll have like a sort of orangish tinge to their skin. So you would see absorption of the light in this region by those types of molecules. If the light that isn't absorbed in the stratum corneum, and a lot of it isn't, you would see go down into the epidermis, and you would have scattering that could occur when the light interacts with structures in the epidermis. You would also have absorption take place, and this is especially true of when you get to the lower layers of the epidermis, such as in the stratum basal layer, where you have melanin present, because the melanin would, of course, absorb light. So you could have scattering, and the scatter could make it go down further, deeper into the skin, into the dermis. Some of the scattered light could actually go back up and be remitted from the surface. You could have light that's down in the dermis be further absorbed by different structures in the dermis, and then it could be scattered. For example, collagen would tend to scatter light, and that would be sort of sent back up to the surface as dermal remittance. So we're going to talk about different types of light that interact with the skin. And this is an electromagnetic spectrum that you can see ranges from gamma rays to X-rays, and then to the UV light, shown here in the three different forms: UVC, UVB, and UVA, and then also visible in infrared light. And historically, UV light has been the light, or the type of light, that most people working in our industry have been concerned with, because it is the most damaging light source. Just as you can see from this chart, we have UVC, which fortunately this is absorbed by, or kept out of our atmosphere by, in the stratosphere we have the ozone layer, which prevents this from entering the terrestrial Earth's surface. But the one that we usually have the biggest concern for is UVB light, which is from 290 to 320 nanometers. This basically causes sunburn. It can also cause changes in the pigmentation of the skin, so immediate pigment darkening, for example, or several other things can happen, resulting in changes in pigmentation. And UVB light also is responsible for skin cancer and photoaging. But as you can see here from this chart, UVA, which is from 320 nanometers to 400 nanometers, is also responsible for pigmentation and skin cancer and aging. And the difference is that the mechanisms between how UVB works and UVA works are somewhat different. UVA tends to operate by radical type mechanisms, causing free radicals to be produced. And for example, UVB light would cause cross-linking of DNA, whereas UVA light would cause oxidation via the production of a free radical species to the DNA. Photoaging, while this can occur from both types of light, UVA is thought to be very highly responsible for that. As you can see here on this graph up above, window glass actually blocks UVB. So if you can imagine, if you were in a car, while you might be protected to some extent from UVB light, the UVA light would actually pass through the window, and you could experience skin damage in that manner. Most of the modern day sunscreens are developed for both UVB and UVA light. They use oftentimes a mixture of different UV absorbers that absorb in both the UVB and UVA region. Okay. And then once you get beyond 400 nanometers, then we're talking about the visible region of the electromagnetic spectrum, and that extends from 400 nanometers to 700 nanometers. And then above 700 nanometers, we basically have the infrared region, and mostly what most research is concerned with in terms of infrared light is the near infrared light, which basically means it's near the visible region. So I'll give you some ranges on that in a minute. So one thing to consider with all these different types of light from the various regions of the electromagnetic spectrum is that they interact differently with the skin. For example, UVB light, which is the highest in energy, but has the lowest or shortest wavelengths actually penetrates to the least extent, only reaching to the bottom of the epidermis. So around the stratum basal layer, that's the limit of its penetration. UVA light, on the other hand, can penetrate to the lower layers of the dermis. And visible and infrared light, you can see, extend into the subcutaneous region of the skin. So, it's important to keep that in mind, because that can give you some type of understanding of what type of cellular structures and molecular structures that these types of light will interact with. For example, UVB light probably isn't going to have as much of an effect on structures that are present in the dermis, whereas UVA light is going to affect both epidermal and dermal structures. So when we talk about chromophores in the skin, again, if these are molecules that absorb light, and usually when a molecule absorbs light, something is going to happen. Some type of change is going to occur to that molecule, and that is normally going to influence the homeostasis of the skin. And just to give you an example, we have urocanic acid here. This scale shows you from 200, so this is beyond, this is UVC light down here, which we're not really concerned about because that doesn't reach the Earth's surface. But once you get up to 280 to 290 in this area, you can see all of these molecules that are shown on this graph have absorbance starting or in that region. And while it's only the shoulder of these peaks, it's still a significant amount, and it will affect those. So for urocanic acid, for example, we know that it absorbs light and it changes from the trans form to the cis form, and when it does that, it undergoes some other procedures or processes, and it ultimately affects the immune system. In other words, it impairs the immune system. When we talk about DNA, if it absorbs, it directly absorbs UVB light, as you can see on here. It doesn't really absorb UVA light, but it absorbs UVB light, and that actually results in cross-linking between adjacent DNA bases in the DNA molecule, or adjacent, I should say, nucleotide bases in the DNA molecule. Just as a sort of side reminder here, well, the UVB light is the light that is absorbed by this. UVA light also causes damage to the DNA, and that's because the UVA light generates free radical species, which then can oxidize the DNA. So it's through an entirely different mechanism, as you can see. We also have tryptophan, tyrosine. These are amino acids that absorb light. Another one is phenylalanine, which isn't shown here, but it's also absorbs light in the same region. And our concern with these types of molecules is that these are amino acids that are part of protein structures, and when they absorb light, they're basically going to form degradation products, and it's going to change that amino acid in the protein structure, and essentially the protein is going to lose its function when you do that. Okay, so if you target amino acids, the target is proteins, and essentially you're damaging the proteins and rendering them to be dysfunctional. We also have the absorption spectrum for melanin that's shown here, and you can see basically melanin starting deep into the UV, in the UVC region, has this sort of monotonic decreasing absorption spectrum that extends well into and actually to the end of the visible region. So it has a very broad spectrum coverage or absorption. So you can see where melanin, it serves as a protective agent against, UV light being absorbed by DNA, trans-Urocanic acid, and even the amino acids tryptophan and tyrosine. Some of the other chromophores that are important in skin, hemoglobin is one of them. And hemoglobin, as you know, is basically a transport protein that transports iron from lung tissue to muscle tissue in the body. It gives the skin, the blood that sort of characteristic red-brown color. But what you'll notice is that if you have hemoglobin in its regular state, that's what gives the red-brown color, which is shown here in the absorption spectrum. And if you have really light skin, you can kind of see the effects of that. And if you look at deoxygenated hemoglobin, oxygenated hemoglobin is this one on the left that gives you the red-brown type color. The deoxygenated form of hemoglobin is the one on the right, and that gives you more of that bluish type color. And again, you need someone with very fair skin to be able to see that bluish type color. But whenever you do see that, you know that you basically have high levels of deoxygenated hemoglobin present. Bilirubin is a similar type molecule in terms of its structure. This is essentially a molecule that is associated with jaundice, which is sort of a yellowing of the skin when you have liver damage. It is one of the chromophores that are present in skin. And then again, we can see the melanin absorption spectrum here as it monotonically decreases. And this is the visible spectrum, so we're from 300 to 700 now. And you can see that this continues to decrease throughout. We also have a little bit of the infrared region that's shown here from 700 to 1200. You can see there still remains melanin absorption. And please note that the scale for this section over here is blown up so that you can see better. But these absorption spectra would actually be like way down here if they were plotted on the same scale as this one over here. You can see the difference between hemoglobin in its normal state, in the deoxygenated state versus hemoglobin in its normal state. And again, the melanin just keeps decreasing in terms of its absorption spectrum. Some other chromophores that we want to keep in mind are these sort of pyridine type structures. And don't really worry about the structure on the left and the structure on the right. The main one to be concerned with is the one in the middle. This is for desmosine. This is a cross-linking agent that keeps elastin and collagen type structures cross-linked together, and it provides them with, or helps provide them with mechanical strength and elasticity. And it also happens to absorb light, and this can influence things. If this absorbs light, it could become damaged, and then you could end up with some type of damage occurring, such as photodamage occurring to these sort of structural protein motifs. We already talked about trans-Urocanic acid. Here is the structure for that. And you can see we have basically 2 nitrogen atoms in this 5-membered ring. And then you have this alkyl chain here with 1 degree of conjugation, and then it ends with the carboxylic acid group. In this case, relative to the double bond here, it's in the trans conformation. But then if this shifts down, as it does in this case after exposure to UV light, you have cis-urocanic acid in this case. And just to show you the difference, here we have the absorption spectrum of trans-Urocanic acid. And you can see, now again, we're concerned UVC is being filtered out, so mostly this to the left here of 280 we're not that worried about because it's filtered out by the atmosphere. But to the right of 280, you can see a significant amount of absorption takes place. So this can absorb light in the UVB region, no problem. After this is converted from trans to cis, you basically have an absorption spectrum that looks like this. So you essentially lose, or the molecule loses its ability to absorb light after it's been converted to the cis-Urocanic form. So let's talk about some positive influences of solar radiation. And in the skin, you have a molecule known as 7-dehydrocholesterol. It's basically a derivative of cholesterol, and it's converted by UV light into what's commonly known as vitamin D3. Vitamin D3 is then transferred via the bloodstream to the liver, and then, so that's one source of vitamin D3. Another source can be dietary intake, where you have, especially from fish and meat products, and that can manage to migrate to the liver as well, regardless of the form, whether it's from the light or if it's from the dietary intake. The liver basically metabolizes this, forming 25 dihydroxyvitamin D3, which is then sent to the kidney, and then the kidney producing 1,2,5 dihydroxyvitamin D3, and this basically results in the increase in calcium absorption in the small intestines. It also increases urinary calcium resorption as well as bone mineralization. So you can see here that vitamin D plays a crucial role in ensuring that ensuring skeletal health is maintained in balance by basically via calcium absorption. So this just gives you some of the molecular views of things. So this is 7-dehydrocholesterol, also known as provitamin D3. Exposure to UV light results into the formation of previtamin D3, and then you eventually have vitamin D3 just due to changes that occur in the presence of temperature. The vitamin D3 form, which is again shown here, this is the same one on the previous slide on the right. So this is still in the skin structure. The vitamin D3 actually binds with this DBP, which is known as it's vitamin D binding protein. This then carries it through the bloodstream and to the liver. Okay, so that was a benefit of UV light, the production of vitamin D3. Now let's look at what happens to DNA when you expose it to UV light. And the most well-known mechanism, which has been understood, is due to the absorption of UVB light by the DNA. And you can see here, you essentially have two adjacent bases in DNA. So you have, this is the backbone of the DNA structure. Then you have like a sugar here, and then this is just showing you the bases, two adjacent bases. These actually happen to be thymine residues or thymine bases. And upon exposure to UV light, you can have two different possible cross-linking type pathways. One is known as cyclobutane thymine dimers. Basically this line that goes from, these two lines that are shown here are basically covalent bonds that are formed between these two adjacent thymine molecules. You can also form one where you have the carbon at the 6th position here, the carbon at the 4th position on this thymine residue, basically forming one single bond, and this is known as pyrimidine 6,4 pyrimidone photoproduct. So UVB light results in the formation of one of these two structures in the DNA. And either one, both of them, these are considered to be mutations of the DNA. So when the DNA tries to undergo transcription to the mRNA is going to be damaged. So it's going to have basically an incorrect sequence, and that's ultimately going to result in an incorrect sequence during translation when you go from the tRNA to the protein structure or the peptide structure. This next slide is actually sideways, so just try to bear with me with this, please. But this shows you the photosensitization of a chromophore. So far, when we were talking about DNA damage, we were discussing UVB light, and now we're going to talk a little bit about UVA light. And UVA light, essentially what happens is we're just labeling this as S. S stands for sensitizer, and again, that can be all different types of chromophores that are present in the skin. That's why we were discussing chromophores earlier. So you would have the chromophore in the presence of the UV light. Basically, the chromophore would enter into this excited state, which you can see here. Then you would go through a process known as intersystem crossing. I don't know if you remember that from either physical chemistry, or you might have studied it a little bit in analytical chemistry. But it's essentially a process that excited state type molecules undergo. Instead of just relaxing back down to the ground state, they go through this intersystem crossing mechanism, and they enter into this sort of triplet state that you can see here. And again, this is still an excited state. So from that point, this sensitized molecule can then undergo either a type 1 reaction or a type 2 reaction that's shown up at the top. The type 1 reaction involves the transfer of an electron, and you end up damaging guanine in the process. So guanine, again, is one of our nucleotides, or one of the nucleic acids. As you modify that, or as you damage that, you're going to damage that link in the DNA structure. If we undergo a type 2 reaction, this is in the presence of, this is normal molecular oxygen. If you remember, normal molecular oxygen has a 3 here. And then from that type 2, you can have 2 possible further reactions take place. Basically, you can have the formation of singlet oxygen, which, again, as you know, singlet oxygen can cause, it's actually a reactive oxygen species, and that causes its own issues that are related to free radical mechanisms. Singlet oxygen can result in guanine modification or damage to guanine, or you can have the minor type 2 reaction that results in this sort of singlet oxygen anion form that eventually results, this is superoxide anion that's shown here. This superoxide anion can be formed, ultimately resulting in the formation of hydrogen peroxide, which in the presence of copper can cause DNA strand breaks, or in the presence of iron can result, and this is the Fenton chemistry again that we discussed in the last chapter, or 2 chapters ago, and this can result in the production of a hydroxyl radical species, which then directly reacts with the DNA and causes strand breaks in the DNA structure. So again, guanine is a molecule that is targeted by UVA light in this whole process that we're talking about here. This might be a little bit hard to visualize, but this is just showing you a deoxyguanosine type molecule on the left. So this is the guanosine base that we're used to seeing. Notice in this position here, you essentially have a normal structure attached to the 5-membered ring here. But then after these reactions take place, like the ones that I showed you on the previous slide, you would end up with what is known as this hydroxyl structure. So just a hydroxyl group. So this becomes 8-hydroxy-2-deoxyguanosine. All it does is it adds a hydroxyl group to the structure of this, and that's enough to basically render the DNA molecule mutated or not functional in its normal state. So UVA light causes damage to the guanosine nucleotide via a free radical type mechanism. UVB light, on the other hand, causes direct damage to thymine residues in the DNA, causing cross-links between adjacent thymine residues. And you have two types, either the cyclobutane dimers or the 6,4 pyrimidone type products. So let's look at the solar spectrum of radiation reaching the Earth's surface. And essentially what we see, if we look at the intensity is shown here. This is the solar spectrum. We see that basically the least amount of light being absorbed at the terrestrial Earth or the Earth's surface, the least amount of light being absorbed is around 290, and then this increases as you increase in wavelength. So you might think, well, you have not as much UVB light being absorbed, but we have to remember that the UVB light is much higher in energy than UVA light. So UVB again is from 290 to 320, and then UVA is from 320 to 400. But one thing to note is, UVA light should not be underestimated either, because there is so much of it reaching the Earth's surface. A sort of nice thing to look at in conjunction with that is if you look at the erythema action spectrum, and what that means is like how erythema is actually formed. You can see that in the lowest part of the UVB region here, the highest amount of erythema is formed, and this decreases as you go or increase in wavelength. So when you get to the UVA region, you don't have erythema being produced nearly to the extent that you do in the lower region of the UVB region. So we keep mentioning UV-induced immunosuppression, and this is essentially a summary slide to just show you different mechanisms on how immunosuppression as a result of UV exposure takes place. So we have several things. This is supposed to be a sun up here with rays coming down, interacting with keratinocyte DNA in the first case, trans-Urocanic acid in the second case, with Langerhans cells in the third case, and then the fourth case is through a mechanism of lipid peroxidation. And essentially in the first case, you have keratinocytes DNA. Again, you have the direct cross-linking with the UVB light that can occur. This essentially causes the production, as soon as this happens in the keratinocytes in the stratum basal layer, you have production of interleukin 10, TNF alpha, and also PAF. So 3 different types of signaling molecules, and these all result in the suppression of the immune system. With trans-Urocanic acid, this is present in the stratum corneum of the skin. When it absorbs light, it's actually converted to the cis-urocanic acid form. And then, it migrates from the stratum corneum into the lower layers of the skin, and then it starts to interact with pathways and mechanisms that are involved with T lymphocytes, which are a type of immune cell that's present in the skin. Actually, T lymphocytes aren't really present in the epidermis, but more into the dermis and into the blood, in the bloodstream. At the same time, we have Langerhans cells that can be affected. If these UV light reaches these, you could have apoptosis occur. You could also have impaired APC function. APC stands for antigen presenting cell. The Langerhans cells are antigen presenting cells, which mean that they sort of they grab little fragments of peptides from pathogens, and these little fragments are known as antigens, and usually they would migrate to T lymphocytes and present that to them so that you could sort of start developing antibodies against that particular antigen. And when you have UV light affecting the Langerhans cells, this could impair their function to do that, and this could result in the Langerhans cells not migrating properly to the T lymphocytes. So you can see that these are sort of intertied together, that cis-urocanic acid is also causing impaired APC function for the Langerhans cell to the T lymphocyte as well. In the fourth case, we have lipid membranes that are present in various cells in the skin. And again, we're mostly referring to the epidermal region. So, this would mostly be talking about the viable epidermis, where you still have, normal cell membrane type cells. This results in signaling and produces this PAF molecule. PGE2 is another one. Don't worry about the actual names of these for right now. If you do need them, I'll get them to you. But you ultimately produce interleukin 4 and interleukin 10. That's really what I'd like you to be concerned with here. That these basically cause immunosuppression. So we can also, look a little bit further, talk about photocarcinogenesis a little bit, and just in general, talking about the development of carcinomas. This just doesn't apply to skin cells, but any type of cell. And just to give you an idea of how this process occurs, so you would start off, these are supposed to be keratinocytes. You would have a mutation that would occur to one of the cells, and again, this could be caused by exposure to UV light. That cell, the cell with the mutation in it is shown here in this sort of different color. Then this whole sort of group of cells would undergo cell proliferation. So you would have proliferation of this mutated cell, so you'd have 2 new cells that are shown here. And then, this would undergo so this is your initial tumor cell population, and then this would undergo a subsequent mutation. And now you can see you have 1, 2, and then this third cell that was shown up here, this is now a different color. So this is, this has 2 mutations in it now. So this is the variant cell at this point. You would have cell proliferation occur again. And again, you wouldn't just have 1 cell proliferation, but you'd have several. And you can see you have the initial 2 cell types that are shown here that are variants. And then you have all these new cells that are produced that are basically variants. So this is the new tumor cell population. You would have another subsequent mutation. Now, out of this whole group here, the one that's shown in red would be the new variant type cell, and then you would have cell proliferation take place, and you would see that you would have all these new sort of red cells that are growing here, and these are going to be growing more rapidly than the other cells, of course. So this is just a general introduction to the development of carcinomas. For skin, the one thing I would like to say, and I don't think we have it noted here in the slides, you essentially have three types of skin cancer that can occur. You have basal cell carcinoma. You also have squamous cell carcinoma. So basal cell, I think you can sort of figure that out. BASAL, and then carcinoma, or cell, and then carcinoma. Squamous cell carcinoma. So basal cell carcinoma, as you would expect, mostly influences the basal layer of the epidermis. Squamous cell carcinoma basically refers to cells that are mainly in the mid-region of the viable epidermis that are being affected by basically the cancer is occurring in that region, and those are the cells being affected. And then of course you have melanoma, and melanoma is really the most serious of these three types of skin cancer, and that's because it can metastasize and spread to other regions of the body very quickly. Here we have an action spectrum for UV-induced cancer, and this is in the hairless mouse. The hairless mouse is a universal model that's used to study influences of solar radiation on the skin. And essentially what this shows is just, it's various experiments that they've done. And essentially, if the higher the value here means the more probability you have of forming cancer when exposed to the various types of radiation that are shown here at the bottom scale. So around 300, that's where you have the peak maximum occur. So you have most of the carcinoma or cancer formed, due to light that is absorbed in this region. You can see that this decreases to a significant extent as you go through the UVB region, and then it keeps decreasing into the UVA region, but then at one point you can sort of see that there's a peak here around 380 nanometers in the UVA region. So this is probably due to the presence of some type of photosensitizer in this type of skin, in the hairless mouse skin, that absorbs basically right around 380 nanometers. Probably producing a free radical species that can then go on to react with DNA structures and subsequently cause the formation or development of carcinomas. So, sort of to end up the chapter, I just want to briefly mention a few things about visible and infrared light on the skin. Historically speaking, visible and infrared light were thought to be innocuous, meaning that they weren't damaging the skin in any way. But now we know better because of recent research findings that have shed light on that. Initial work was done on visible light, and it's seen that visible light actually induces erythema to some extent. It also produces pigmentation in the skin. In addition to that, it causes thermal damage, and of course, it also produces free radicals in the skin. What we also should keep in mind about visible light is that it can activate photosensitizers, and these can be either endogenous species, so things that are present in the skin normally, or it could be exogenous chromophores that are, for example, people undergoing certain treatments might have, various pharmaceutical agents that are exogenous chromophores that are present. But in either event, this can result in the formation of reactive oxygen species, ultimately leading to DNA damage. And then the last item here, of course, is infrared radiation has also been shown to induce the production of free radical species, or free radicals in skin. And there's a lot of research underway in this area right now. So this is where I told you that before, you have a lot of multinational companies that are working on new products that are meant to combat the effects of infrared and visible radiation on the skin. And to do that, they're looking at ways to prevent these free radical type mechanisms from occurring. So that brings us to the end of this chapter, and you can get this chapter is a pretty good chapter in your textbook. So you'll want to cover the relevant sections that we looked at in the slides today, and try to get a good understanding and grasp of what we discussed. And I hope you enjoyed this chapter, and we will talk next time. Thank you.